Welcome to this complete explainer on cell structure and function. Today, we're going to scale way down to the microscopic building blocks of life and then build our way back up to the macroscopic level. If you're prepping for your O levels or you just really want a comprehensive, professional grasp of cellular biology where we cover absolutely everything, well, you're exactly in the right place. Let's get into it.
Here's our roadmap for today. Number 1, we'll meet the cell organelles. 2, viewing cells under microscopes. 3, specialized cells and functions. 4, going from cells to organisms. And finally, number 5, mastering magnification calculations.
All right, section 1, meet the cell organelles.
Okay, I want you to think of a cell like a bustling, highly organized factory. First up is the big boss, the nucleus. This is a 7 micrometer control center. If you look closely, you'll notice a double membrane enclosing it, complete with these little nuclear pores for exchanging materials. Inside, you've got darker bodies called nucleoli, and this thread-like stuff called chromatin, which actually condenses into chromosomes during cell division. The nucleus is absolutely vital because its DNA dictates exactly which proteins your cell's cytoplasm is going to produce.
Next up, we have the mitochondrion. You probably know this one as the powerhouse of the cell, measuring in at about 3 micrometers. It's double membranous, but check out that highly folded inner membrane. Those folds are called cristae. This right here is the exact site of aerobic respiration, generating all the ATP energy your cells need to survive. And here's a really cool detail. It actually contains a fluid matrix housing its own DNA and 70S ribosomes, so it can synthesize its own proteins. How wild is that?
Then we have the chloroplasts, which are basically the solar panels for autotrophs. Now, these 4 micrometer organelles are exclusive to plants. Inside, you'll find these impressive little stacks of thylakoids, known as grana. These membranes contain the green pigment chlorophyll, which literally captures light energy. That energy is then used to biochemically convert carbon dioxide and water into carbohydrates, like glucose.
So let's put all of that together and contrast our 3 main cell types. First, plant cells. They're defined by their thick cellulose cell walls that prevent them from bursting under high turgor pressure, plus they feature a large permanent sat vacuole right in the center. Next, animal cells. They lack both of those structures completely. Simple enough, right? Finally, bacterial cells are just a whole different category. They lack a true nucleus, possessing instead circular DNA and these extra little DNA loops called plasmids. On top of that, their cell wall is made of peptidoglycan rather than cellulose, and they rely on slightly smaller 70S ribosomes.
Moving right along to section 2, viewing cells under microscopes.
So to actually see these tiny structures, we need to know exactly how to prepare them in the lab. Let's walk through how you'd prep a plant cell slide using a simple onion. First, you peel a thin piece of epidermal tissue using forceps. Second, carefully lay it perfectly flat in the center of a glass slide. Third, and this is key, you add a few drops of iodine solution. Why? Because iodine stains the starch in the cells, making the different structures pop. Finally, using a mounted needle, you support a cover slip and lower it gently at a 45-degree angle. That angle is super important to avoid trapping any of those frustrating air bubbles.
Seeing the breakdown of that methodology really brings it to life, right? Peeling the epidermis, placing it flat, staining it, and that crucial 45-degree needle technique. I highly recommend memorizing these exact sequential steps because examiners absolutely love to test this in practical methodology questions. Now, shifting gears to animal cells, we typically look at human cheek cells. Since they're really soft and flexible, we just lightly swipe the inside of the cheek with a clean cotton bud and smear it onto a slide. But instead of iodine, we use methylene blue dye. You see, animal cells don't have starch to stain, so the methylene blue reacts perfectly to make the otherwise nearly invisible nucleus stand out loud and clear.
All right, on to section 3, specialized cells and functions.
In complex organisms, cells undergo a division of labor. They develop distinct shapes to do one crucial job perfectly. Take the nerve cell, or neuron, for instance. It features these incredibly long fibers designed specifically to conduct electrical impulses rapidly across vast distances, literally from your brain all the way down to your foot. But in stark contrast, look at red blood cells. They're these tiny biconcave discs that actually abandon their nucleus entirely when they mature. They just ditch it. This amazing adaptation maximizes their internal volume so they can pack in more of the red pigment hemoglobin for optimal oxygen transport.
And plants? Well, they have their own highly trained specialists too. Root hair cells develop these long, hair-like projections that push right between soil particles.
This gives them a massive absorbing surface area for soaking up water and mineral salts. Meanwhile, way up in the leaves, we find the palisade mesophyll cells. These are tall, columnar cells completely packed with chloroplasts, positioned right under the upper surface of the leaf to trap maximum light energy for photosynthesis.
Section 4. From cells to organisms.
Now, we're going to zoom way out to see how all these individual specialized workers group together to form a highly functioning global economy. We call this the hierarchy of life. It all starts with the cell, the basic structural unit, like a single muscle cell. A group of similar cells working together forms a tissue, like muscle tissue. Then, different tissues working together form an organ. Take your stomach, for example, which combines muscle, epithelial, and glandular tissues. Next, groups of organs with related functions form an organ system, like your esophagus, stomach, and intestines making up the digestive system. And finally, the whole shebang. All those systems working in perfect harmony create a fully functioning organism.
Section 5. Mastering magnification calculations.
Okay, I know math and biology can feel a little intimidating, but trust me, it is incredibly straightforward if we just follow a few golden rules. Let's define the core concept first. Magnification is simply the ratio of image size to actual size. The formula is m equals I divided by A. The most important thing to remember here is the distinction. Image refers to what is printed on your exam paper or the screen right in front of you, while actual is the real-world microscopic size of the biological specimen.
And here is your ultimate cheat code for these calculations, the magnification triangle. With I at the top and A and M at the bottom, it guarantees you will always set up your division or multiplication correctly. Want to find magnification? Just cover the M, leaving you with image divided by actual. Need the actual size? Cover the A, and you get image divided by magnification. It is literally a foolproof method for exam success.
But listen up, because you need to be aware of the single biggest trap in exam questions: mixed units. You absolutely cannot divide centimeters by millimeters. Both values must be in the exact same unit before you even touch your calculator. The easiest trick? If your ruler measures the exam image in centimeters, simply multiply it by 10 to convert it into millimeters. Remember that magic number 10. Align your units, and the math is going to work out perfectly every single time.
Well, we have covered a massive amount of ground today. From zooming into the microscopic inner folds of a mitochondrion to calculating the exact magnification of a lab slide. But before we wrap up, I want to leave you with a thought. Right now, inside you, trillions of these invisible cells are seamlessly executing a perfect division of labor. If a single microscopic cell can house its own dedicated powerhouse, a genetic control center, and a complex transport system, just imagine what infinite potential the whole organism, you, holds. Keep exploring, keep questioning, and I'll catch you in the next explainer.